when the body makes glutathione, we're not talking about your ability to make glutathione. We're talking about your ability to use glutathione. And this is a very important distinction. Again, I hear too many clinicians talking about, yeah, you're not making glutathione very well. No, more often than not, as human beings, unless we've got a really deplorable diet, which, mind you, sadly, is a fairly common phenomenon. But assuming a decently balanced diet, meaning that we do have at least the building blocks of nutrients in our diet, making glutathione isn't the problem. Using glutathione is where there can be a problem. So glutathione, if we had a toxin, and this is glutathione, when we glutathionize, when we bind glutathione onto that toxin, we have rendered the toxin usually less toxic, neutralized, and we have importantly rendered it water soluble so that it can be removed from the body. Keep in mind that glutathione is not only a detoxifying agent, it is an antioxidizing agent. In fact, it is probably one of the most, if not most potent antioxidants in the human body. So we glutathionize toxins and we glutathionize oxidants. Now, as we said, the problem can occur in how efficiently we glutathionize things. There are a family of genes whose very job is to direct, to orchestrate the movement of glutathione enzymatically and to catalyze, as enzymes do, the binding of glutathione to its substrate. That family of genes is known as the GST genes. The glutathione, drop the S. S is a biochemical jargon, so let's drop the S. The glutathione transferase genes. These are the genes making the enzymes that control the transfer of glutathione onto its substrate. There are three important members. There are actually more members to the family, but three of the members are the most important. And these members, these genes of the GST family, are almost identical duplicates to each other, almost. There are slight sequence variations between the individual genes. But the remarkable nature here, Lynn, think about it. If we go back to what I said earlier, that detoxification is one of the most important adaptations of the human body, look at the miracle of this. The miracle is we take, for example, and the GST genes are differentiated by their suffixes. So the three important GST genes are GST T1, theta, GST P1, pi, GST M1, mu. These are the three important, they're called isozymes, almost duplicates of each other. But look at what's remarkable here. The GST T1 gene is found on chromosome 22, volume 22 of your encyclopedia, your genomic encyclopedia. GST M1 is found in volume 1 of your encyclopedia, i.e. showing you that the miracle that these genes, even though they're duplicates of each other, they're spaced out on different parts of the genome. Perchance something were to affect one of these genes, we do not wipe out glutathione function. If you as a human being had no glutathione function, hypothetically, you would not make it past embryonic development. You would be the product, what we call a product of conceptus. You would be a miscarriage. You wouldn't even be able to withstand the mild toxic environment of mommy's womb, much less withstand, development in the real world. Now, in as much as these...
Mansoor, can I just ask for a point of clarification at this point? So
yes.
One thing which I'm sure you've seen is that many clinicians know about these gene deletions. Mm-hmm. I have a gene deletion. Mm-hmm. And what they do is they think, oh, glutathione deficiency, I'm going to give my patient more glutathione. That will fix the problem. As you alluded to, that will not fix the problem.
Oftentimes it would not, and oftentimes it can cause more concerns.
Exactly.
Into long term. So we're going to get there very shortly.
Good, good.